So today we will do a biochemistry test for you. So first of all, this looks like this is a urea test. Lee, take a picture of this one. This is a urea test. So the urea test, we're gonna first of all do the label. So we will put Proteus vulgaris there. So we write the PV, and we write the initial. Take a picture. Again, we do a label, bench number, and date. Okay. Urea is in the liver. It will be break down, become ammonia and carbon dioxide. Okay. So if it's generated ammonia, it should be mild. We call it a weak alkaline. It turns pink color. So let's say we have a Proteus vulgaris on the agar plates. So if we have a Proteus vulgaris here, Proteus vulgaris, we pick some, then we put it inside. Take a picture of this one. That's very easy, the first one. The urea tube. The second one is we will to do a 3-tube fermentation test. So the first one is glucose. We write a G there. We will do E. coli, so we write E. coli, EC, bench number, initial, and date. And the second one is lactose. So we write L there. We also put the E. coli there, and the CS, bench number. And the last tube is sucrose. So we add S there, and we have initial, bench number, bacteria name, and date. Now, very important, when you can see inside. Take a picture of this one. There is a Duran tube inside that's going to test the gas, whether bacteria generate the gas. See inside there's a Duran tube. So the same thing we're going to do inoculation is very simple. We just add it in there. So we're going to do E. coli, so we're going to add the E. coli. So Lee, take a picture of this. And the E. coli. So one more. So we pick the E. coli. Then we go here. And we will be switch, use a new book. Okay, so the second one. Also want to take the E. coli. So we go lactose. And then inoculate to go to the sucrose. So we hope we'll see a bubble there, which is turned yellow color, which means acid fermentation positive. So these are the 3-tube fermentation. Of course, all these need to be incubation. Okay, now the next one, which is called the SIM test. SIM test, which is called sulfide indo and the motility. So here we will do E. coli. So we write the EC there. Of course, the initial name, and then you can write the initial here, bench number and date. And the second one we want to do is Proteus vulgaris. So we write the PV. We're gonna do date, initial, and the bench number. We can write like that. So this is something very special. Instead of we using the loop, we have to using is a needle. Okay, so you're gonna use the opposite side. So let's say we do E. coli. We're gonna pick E. coli from here. For the needle injection, we will be very simple. Is from the beginning to the end is just a one click. Then we take it out. Okay, so we don't use loop, we use a needle. And this one is also the same. We will do Proteus vulgaris. So we use Proteus vulgaris like this. With some colony. And we will do just needle injection, which is right in the middle. And SIM agar is a semi-solid agar, which is containing 0.5% agar. So, okay. Then what we're going to do is a deaminase test. Deaminase test, this is called a phenylalanine deaminase test. So we will write the PHE there. We're going to do Proteus vulgaris, initial date, and also the bench number. This will be a very simple streak plating. And after incubation, we will add FeCl, which is ferric chloride. If ferric chloride turns turns green, which means it is positive. Otherwise, it's negative. So let's do Proteus vulgaris. So we pick Proteus vulgaris right here. And then we do a streak. Okay, the streak is like this. So take a picture of it. So we do streak. Okay, that one is done. Then the last one is the MrVP test. We mentioned the MrVP test is part of the IMVC test. So MR is methyl red, VP is we call it's a Vogus Persky. Okay, so we're gonna label. We will do two, which is E. coli and Enterococcus. Oh, sorry, Enterobacteria. So date and also bench number. Because their results are totally opposite. So this is we add EN, it's ENTA, and Enterobacteria. C0.115 and the bench number. For IMVC test, E. coli is positive, positive, negative, negative, and Enterobacteria is negative, negative, positive, positive. So how we do it is very simple today, but later on we will do a confirmation test. So this is E. coli, we just pick some E. coli. Just pick some E. coli. And then we put it inside of the E. coli tube. That's very easy. And I'll show you confirmation test next week. And then for the Enterobacteria, we also do the same thing. We will add a little bit. And then we will inoculate into the VP broth, MrVP broth. Okay. So these are the sets of the biochemistry test on the tube we finished. Now we want to do two other tests. So first the test which is called a catalase test. So catalase test, we label it. We're going to do split them into two. Left side we do Staphylococcus aureus, right side we do E. coli. And of course you write your name, initial, and the date and the bench number there. This is also we're going to do very simple, do a streak plating. So how do you do it? Very simple, we're going to add E. coli on one side. So take a picture of E. coli. And then we add on one side. Okay, just a gentle streak. Then the second one is Staphylococcus aureus. We got Staphylococcus aureus. And we are very simple to do right here, which is a streak. Now after incubation, when they grow, we will add actually H2O2. That is called hydrogen peroxide in there. If there's a bubble, which is positive. Okay, so this will be keep right here. Now the second test, we call it oxidase test. Oxidase test used to testing Pseudomonas aeruginosa. So on one side we add Pseudomonas aeruginosa, on the other side we add E. coli. Of course you put your name, bench number, and date there. Okay? So the same thing we do a streak plating. Can I have a loop? So we have to prepare two. Is Pseudomonas aeruginosa, another one is E. coli. Okay. This is a very easy and a quick test to identify Pseudomonas aeruginosa because it is a gram-negative bacteria. So they both grow on the Macanchi agar, you're not going to see a difference. So Pseudomonas and we will streak onto the side. And then we'll be using E. coli on the other side. After incubation, we want to see the color change. We all need to add oxidase reagent. If it shows purple, which means it's positive. Now we're going to do the three test, which is for the bacteria using enzyme. These all three tests are very easy. We just split them onto two. Strict plating bacteria, we see where it's grow. Now this first one is a skin milk agar. So this is skin milk agar. On the left side, we're going to add bacillus, and on the right side, we're going to add E. coli. And of course, you're going to add name, initial, and date there. Okay. The second one is a split brood agar. This is test the bacteria whether have lipase. So we also split them onto two. On the left side, we're going to add, this is split blue agar, SBA. So we add SA, right side we add E. coli. This is going to be the initial, date, and the bench number. The third one is the starch agar. We want to see whether bacteria can use starch, has amylase. The same thing, we split them. So we have starch agar. Then we have Bacillus cereus, Bacillus subtilis, and they have E. coli. So we're going to write the name, initial, and the bench number. Okay, so very easy, which is we're going to do the inoculation. And how to do it, we do these each one by one. So first of all, we get E. coli. Let's say we get E. coli. Okay, so we're going to put right here, we're going to streak plate here, right here. Then we're going to get bacillus. So make sure you get the bacillus. Then streak plating on the right side. Okay, then this is done. Second one is split blue agar. So we're going to inoculate E. coli and the Staphylococcus aureus. So we do Staphylococcus aureus. We do the right side. And then we'll have E. coli. So we get E. coli. Then do this on the left side. Now, the last one is the starch agar. We go back to see bacillus and the Staphylococcus aureus. So when we do it, oh sorry, bacillus and the E. coli. So we have E. coli on one side. And the bacillus on the other side. Okay, now all these will do the incubation, we'll see the color change. So, thank you.